to hear our next series of talks. We're going to continue with our first group of preclinical optical imaging, and it's really a pleasure that I have now to introduce my good buddy and colleague, Dr. Bill Ross. Dr. Ross is a professor in the Department of Physiology at New York Medical College. Like several people here today, he trained in high energy physics to begin with, but we're not going to go too far into that question. You can read his biography along with others on the website after this is over. He's a professor in the Department of Physiology. He's done path-breaking work on particularly with wide field microscopy. And today he's going to talk to us about high resolution imaging of sodium and calcium changes in tiny little dendritic spines.
Thank you. Okay. This is great, this idea of imaging symposium, bringing people together of different perspectives and interests in this field. And I'm going to take the sort of talks that we've had so far, which have been at a clinical level and a tissue level, and sort of bring them down to the subcellular level, little microscopic pieces of tissue on dendritic spines. And also we're going to have a different temporal perspective. What we've seen so far have largely been pictures, but there's no time in what we've seen for the most part. And I'm going to be showing you dynamic changes in cellular properties, largely using indicators that can respond rapidly to changes in sodium and calcium concentration changes. And I'll go into more detail about that as we go along. So first, let's see. Okay, so in the nervous system, the vast majority of synapses that are made on cells are excitatory synapses are made onto dendritic spines. Each neuron in the brain, for the most part, has thousands of inputs onto it. And these are just a few of them, sort of showing where the connections are made onto the inhibitory synapses are not usually made onto spines, but excitatory synapses are made onto spines. Spines are very small. Here is a light microscope picture of a cell that's been filled with a dye to show some structures. And you see that there are spines. The scale is very small. This is 1 micron. So typically these spines are in the order of less than a micron in length. So if you want to understand synaptic integration, it's a very fundamental process of what goes on in the nervous system. You want to know what happens when individual inputs, action potentials coming along these presynaptic fibers. What do they do? But it's hard to actually make recordings at the target because these spines are so small. So what people have done, and which we are in that tradition, is we've tried to use imaging techniques where we try to reveal information about what goes on there by finding a transducer that can tell us something about what's going on there. So what are we interested in understanding about synaptic integration? One is when a synapse is made and with presynaptic cell releases glutamate onto the postsynaptic targeting cell, it's going to generate an EPSP. We'd like to know in detail how that EPSP is generated and then how it spreads to the cell body of the cell, okay? And also, we've learned from many other experiments that in addition to generating a voltage change in this postsynaptic cell, there are calcium changes and other things as well, but in particular calcium changes that are made that in turn lead to the changes in the properties of the cells, for example, long-term potentiation or long-term depression, which are as a consequence of the calcium changes in these cells. So we'd like to understand that better. So we use these optical methods, as I said, to tell us something about that. And what we're interested in is how the different channels in the cell are activated and what happens when the glutamate acts on the receptors, the AMPA receptors, the NMDA receptors, and then there are other channels in the membrane. So we'd like to follow the sodium that flows through the AMPA receptor and possibly through the NMDA receptor. We'd like to follow the calcium that goes through the NMDA receptor. Generally, calcium does not go through the AMPA receptor, so we don't follow that. And we're also interested in activation of the voltage-gated calcium channels and voltage-gated sodium channels. So if possible, we'd like to use imaging to tell us all these changes when action potentials release glutamate onto these spines. Okay, so among the questions of some interest are, when we make these measurements, is there a regenerative component to the spine EPSP? And typically what most people have done in electrical recordings from these cells, mostly from the cell body, they can't tell what's actually going on in the spine, but there's been hypotheses that have suggested that there are action potentials actually generated and restricted to the spines. And could we see that in the cells? Okay. And then there's this, let me go back one. Yeah. So in addition, does this spine neck resistance insulate the spine from the dendrite? And finally, what are the properties of the calcium change that cause influence in downstream events like LTP? Okay. And so, as I said, we've used imaging techniques, and in particular, we've sort of pushed certain parts of the imaging forward, and that's sort of been our distinguishing characteristic, is using some advanced imaging techniques to examine these things. So what have we done? First, for calcium changes, many of you are probably familiar with indicator dye like Fura. It's a well-known calcium indicator dye. One of the nice properties of Fura as an indicator is that it's very sensitive to the calcium change because its KD is right in the range of the resting calcium in the cell. And this is great for making it a good detector of calcium change, but it's not good if you want to follow the time course of the calcium change, because an indicator that has a KD at the resting calcium level is going to be a buffer. Okay, so in order to avoid that buffering, we use less sensitive calcium indicators with about 100 times less sensitivity. We still can detect it, but because it's 100 times less sensitive, it doesn't distort the time course of the calcium change. And that's what we're interested in. We're not just interested in detecting the calcium change. That's been known for a long time. We're interested in measuring its dynamics accurately. Okay. The second thing is we'd like to detect the sodium changes. As I said, sodium goes through most of these channels and receptors, but no one's ever measured the sodium coming through these channels. So we've used a sodium indicator dye, which has been known around for a while, but no one's ever pushed it to the point where you've actually able to detect sodium changes in individual spines. And so that's one thing that we've done, and I'll show you those results. Okay. And the last thing from a technical point of view is that we were interested in sodium changes and calcium changes, and we've developed techniques to measure them both simultaneously at high time and spatial resolution. And I'll show you how we do that in the next couple of slides. So here's a typical experimental setup. We make brain slices, as you've already heard, cut the mouse brain like on a cutting salami with about a thickness of a millimeter, put it in a dish, and we're going to have two electrodes, and we're going to image it under a microscope. And after we patch the cell with a patch electrode, and the electrode contains the sodium indicator dye and the calcium indicator dye. And after we patch it, the indicators can spread throughout the cell, and we can image that calcium, those indicators, and we see the morphology of the cell. but also, since they're indicators, they're going to tell us something about how the sodium and calcium changes when the cell is stimulated. Okay. So this is a hippocampal slice. These are the cell bodies. And so here's the patch electrode on the soma. And that, in addition to putting dyes into the cell, this electrode can record the potential in the cell and can also stimulate the cell to fire, among other things, can stimulate an action potential. And it's been known for a long time that action potentials not only spread down the axon up this way, but back propagate into the dendrites like this. And the second electrode is used to stimulate the presynaptic fibers to cause a response in the dendrite. And we're going to then image a very small part of the dendritic field with a high resolution camera to tell us what's going on in the spines, which you can't see in this figure, but which will blow up and you'll see in the next figures. Okay, so this sort of picture looks a little funny because I'm going to fill up the screen with other things. Okay, but here's a piece of a dendrite, and when we're going to stimulate it, okay, and the way we stimulate it is the electrode in the soma is used to stimulating this back propagating action potential, and it not only can it stimulate it, but it records it. This is a recording in the cell body. And then the stimulating electrode can stimulate a synaptic response, and then a second synaptic response, which is super threshold for generating an action potential. So then we're going to take an image of this field with both the calcium and the sodium indicator dyes, and we're going to take the difference image between a point before and after the synapse. So this will tell us the change in the sodium and calcium change when this synaptic stimulation is made. Okay, and what we'll do, and we'll look to see where in the dendrites, if at all, we've generated this change. And what we see is that not all the time, but some of the time, we'll see a very localized, on the order of 1 micron or less, a change in the indicator dye that reports calcium, and at the same time, a change in the indicator dye that reports sodium. So both of these are activated at the same time. So then, knowing where these changes occur, these are at the same location, we can put a region of interest marker on the dendritic segment, and we can then look as a function of time to see the changes in sodium and calcium change. Now, the sodium looks like it's going down. That's just because the indicator dye reduces its fluorescence when sodium goes up. These are actually increases in sodium. Okay. So what you see is simultaneously we're measuring both sodium and calcium when this stimulation protocol is generated. You can see that even though you can see a calcium change from the back propagating action potential, but no sodium change in the dendrite. But synaptically, we see a very clear sodium change. So this lack of sodium change here from the back propagating action potential is not because of a lack of sensitivity. We can prove that in a number of different ways. It's because there are very few sodium channels in the spine. So that's one conclusion from this result. On the other hand, we see that the synaptic response generates clear sodium changes, also generates calcium changes. The time course of the two look about the same, but they're actually due to two different mechanisms. And it's actually fortuitous that the rapid recovery rate of sodium and calcium is about the same. Okay. So then we put on a classic synaptic blocker, APV, which is a blocker of NMDA receptor channels. And what we see is that you don't see much change in the electrical response, and that's because most of the electrical response is generated from activation of the AMPA receptors. And we can see that because the sodium change does not change at all very much when we put on APV. On the other hand, the calcium change is almost entirely blocked, the synaptic calcium change. Okay, this remaining calcium change is due to the action potential and not due to the synaptic response. Okay. There is a little bit of a calcium change. That's due to voltage-dependent calcium change signals. So very clearly here we have some clear results. No, go back. No voltage-dependent sodium channels in the spine. Sodium mostly through AMPA receptors, not NMDA receptors. Blockade of most of the calcium response synaptically, but not from the voltage-dependent signal. Oh, I didn't know I was. All right, I'll go fast. Okay. So how is sodium removed from the spine? Okay, it's removed, unlike the calcium, which is removed by a pump or buffering into intercellular compartments, the sodium is removed by diffusion from the spine into the dendrites. So here's, you look at the sodium change, you see it's at the spine, it's fast, but you actually can see it in different places along the dendrite, but it's getting slower and smaller. The calcium change, you don't see it, you only see it in the spine because it's calcium is buffered, it diffuses 100 times slower than the sodium. Okay, let me go quickly through this. Okay. Okay, so the main conclusions from the sodium part of this experiment is that most synaptically activated sodium entry is through AMPA receptors. Entry through voltage-gated sodium channels and NMDA receptors is small. And sodium removal, and this I didn't go into in detail, sodium removal from spines and other isolated structures is very fast, primarily by diffusion. Okay, so let me just quickly mention a couple of calcium results. I don't have enough time. It looks like the calcium signal from the voltage backpropagating action potential and from the synaptic response is very similar, but actually they're different. And the half decay is slower than from the synaptic compared to the backpropagating action potential. And the reason for that, and here's this, I'll just skip right to this point, is that even though it's known that the synaptic response of the NMDA receptor from glutamate pulse is slow, it's known that this is only when the magnesium component of the solution is there, and the synaptic response removes the magnesium, allowing calcium to flow through the NMDA receptor. So it's only when the magnesium block is removed that you get calcium entry, and that occurs only at the time of the EPSP in the spine, which is very fast. So I will skip to the end, and at this point, so what do we know about the calcium signal? Okay, it's very fast, but not quite as fast as from the backpropagating action potentials. And the fast time course of calcium change is consistent with most calcium entry coming through NMDA receptors when the magnesium block is removed by the fast AMPA-mediated EPSP. And a smaller component, and this I didn't have a chance to go into, comes through the NMDA receptor at rest through voltage-gated calcium channels. And finally, the fast calcium transients imply that the targets must react quickly. Okay. Thank you very much.
We have time for one question. Is there a question from the audience? If not, I will ask you a question. Okay, I'll ask you a question.
Okay.
So yeah, you said that you showed beautifully how spatially restricted the calcium is at the site of the spine and doesn't diffuse into the shaft of the dendrite. But you said it's because it diffuses much more slowly.
The calcium, that's
Yes.
right.
So what is it that restricts the diffusion from entering the shaft of the dendrite? Is this just buffering mechanism, or it's a
It's
pump?
buffering. I mean, That's right. I mean, the cytoplasm in the neck is not that different from the point of view of diffusion of ions, not that different from any other intracellular compartment. So if we did a computer model, and I had one there, but I skipped over it, and you look at how sodium or calcium diffuses, it's 100 times slower than sodium, but it goes in there eventually. But on the time scale of what I was showing, not much gets into the dendrite.
So is the implication of that, though, that some of the calcium signal that enters into the dendritic spine with synaptic activity could actually be affecting changes through action, not only in the spine, but also in the proximal portion of the dendritic shaft?
Well, it's pretty complicated. So first of all, two things that I didn't really emphasize. One is there are voltage-dependent calcium channels, and the voltage actually spreads from the spine into the dendrite pretty well, which again, I didn't have a chance to show. And since the voltage spreads there, it can open up the voltage-dependent calcium channels in the dendrite, and that'll cause a calcium change there, okay? And the second thing is that if a single synaptic stimulation from a single presynaptic fiber will cause the responses that I showed, but if you have multiple inputs coming at the same time, you can get a regenerative response that they call an NMDA spike, and that can spread well into the dendrite. So that's another way you can get a calcium change in the dendrites. But from a single fiber, the calcium change is generally restricted to the spine.